ever felt someone staring at you? Raise your hand. Yeah, me too. And sharks and fish can also feel the same sense. So they have this thing called the lateral line that's in their bodies, as you can see over here, and it's found in most sharks and fish. And it's the sensory system that detects currents and pressure in the water, so it can detect prey and predator that's swimming around them. And something that's really cool about the lateral line is that it's comprised of these little things called neuromasts. This is a zebrafish. And the neuromasts are these sensory organisms found within the lateral line. And the neuromasts are comprised of 3 different cells. The mantle cells, which you can see are these green ones over here. And those kind of serve the purpose of support or structure for the neuromast. And then there's the support cells, which serve the purpose of offering the signaling and the growth and development that the hair cells, the actual sensing cells, need to do their job. And so, these are hair cells over here. And something that's really cool about these hair cells is that in zebrafish, they are able to regenerate, and in humans, they are not. And so in humans, the hair cells are very similar to the cells that are found within our inner ear. And so when they're damaged in humans, they can lead to deafness and other hearing problems, but in zebrafish, they can regenerate. And so a lot of the steps in kind of focusing about where to go in this research field is to look at what is causing these neuromasts, these cells to generate. And so that's the big picture question for this presentation. And the journal that I'm looking at today is called J&K Inhibition Inhibits Lateral Line Neuromast Hair Cell Development, or in simpler terms, hair cell generation in the zebrafish lateral line. Old zebrafish. And so I'm specifically looking at J&K signaling. And so J&K signaling is involved with a lot of development and growth in the lateral line. And so, and then the SP600-125 is an inhibitor of J&K, so that means that it stops the J&K from doing its growth and development. And so, as just like a little diagram to kind of give you guys an idea about what happens. So the J&K leads to this hair cell gene expression, and the SP600-125, the J&K inhibitor, stops this from happening. And so the scientists want to kind of look at how is this SP600-125 actually working? Does it actually work within this as a J&K inhibitor? Because they've done some background research, but they want to know, does it work? And so something to know as we go onto this slide is the FM143FX, which is also right over here. And basically what that does is that it's just a dye that marks off, do the cells actually work? And so these cells over here show that they are doing the sensing, the sensing on the lateral line. Yes?
Yeah, what kind of protein is the J&K protein? Is it like a receptor, or where is it?
It is found with the SOC2 cells over here, and it's found, like when you look at the neuromast, it's found within those supporting cells that are over. So it's found over here.
So inside them.
Yeah. And so what they also are looking at are dose dependence amount of this SP600-125, which is the J&K inhibitor. Yep.
Do you know what that dye stands for?
With this dye
showing functionality.
Yeah. It just shows, is it actually doing the sensing that's like activity? Is it actually doing the sensing that's happening with the cells? And so they use increasing amounts of SP600-125, and this is just the control. So that means that these are just the normal cells doing their own sensing, their own functioning. And what they find is that there's less functionality as there's more of this J&K inhibition going on. So that means that if we go back to what I was talking about before with this diagram, can anyone explain to me what SP600-125 is doing? Yes?
Inhibits J and K.
Yep, it inhibits J and K, and what does J and K do?
Activate the
Yeah,
regeneration.
the hair cell gene expression, yeah. And so, yeah, it inhibits the hair cell gene expression. And so now that they know this, they want to look at it a little bit more specifically and look at what part of the neuromast is it actually targeting. And so what they're doing is taking immunofluorescent dyes, so they're just staining different cells to look at what's lighting up, like what's actually doing it, what's functioning. And so they also look at this with dose-dependent amounts. And something else to just keep in mind when we're looking at this is the S phase, which is in the cell cycle. It's the synthesis phase of the cell cycle. And it's, as you can see over here, it's what is happening when the DNA is replicating. And so just keep this in mind when we're going into our next one. So what you can also see, this is kind of similar to the graphs that we looked at before, but as you increase the SP600 125, and each one of these parts of the hair cells, so the actual neuromast hair cells, the supporting cells, and then the proliferating cells, they are all going down as you increase the SP600 125 or the J and K inhibitor. So that means that J and K plays a role in each one of these parts. And so, does anyone want to tell me what SP600 125 is doing with all of these? Keep in mind that S phase is really important for this one. No? Okay. So this means that the SP600 125 is having fewer cells progress into this S phase of the cell cycle. And so, as you can see earlier, what's happening is that these cells are losing their function. So that means that if less cells are going into this S phase of the cell cycle, that means that there's less cells being produced. So more cells are going into cell cycle arrest. And so that also shows that SP600 125 is working. And so the third step is that now that they know that it targets this cell cycle arrest, they also want to look at cell cycle death, so apoptosis. And what they look at specifically with these graphs is this caspase-3. And caspase-3 is kind of an indicator of apoptosis or cell death. And if there is more caspase-3, that means that there is more cell death. And so they also look at this with dose-dependent amounts. And what they are looking at is that as you increase this SP600 125, they find that there are more caspase-3 cells in the neuromast with both of these graphs. And so what this means is that there is going to be more apoptosis within these cells. And so that means that there's also not only cell cycle arrest, but there's also more cell death happening with this SP600 125. And so does anyone want to tell me what caspase-3 is doing with SP600 125? Yeah.
There's more apoptosis.
Yeah. Yeah, so there's more apoptosis or cell death. And so they kind of take this another step further, and they want to look at specific proteins that are involved with this cell cycle arrest and apoptosis or cell death. And so what's happening is that they're looking at P21 and P53. And so P21 is involved with the cell cycle arrest, and P53 is involved more with apoptosis or the cell cycle death. And they want to look at this in regards to J and K. And so they know that P21 and P53 are related to J and K, but they want to find out specifically if one protein is favored more than another, or they want to kind of get a sense about that. And so what they're also doing, the kind of protocol that they're using to find this P21 and P53 is called whole mount in situ hybridization, which takes specific tissue slices or parts of the zebrafish and then expresses all the genes in them to find out what's happening in them. And so, as you can see over here, these are the controls, and then this is the SP600 125, the J and K inhibitor. And what's happening is that they're looking at the amounts of P21 and P53 within J and K. And so what they find is that there's not only cell cycle arrest with this P21 because there's more SP600 being produced, but there is also more cell death or more apoptosis happening, which means that SP600 125 is both affecting apoptosis and it's both affecting the cell cycle arrest that's happening. And so, as you can see, J and K inhibition is affecting P21 and P53, which is leading to the cell cycle arrest and cell cycle death. And not cell cycle death, and cell death. And what's really happening, or what this means, is that J and K plays an important role with the way that the cell cycle moves and the way that the cells kind of continue to reproduce. And so, the next step is looking at J and K and its role in regeneration, because now that we know that J and K plays an important role in development and how the cell cycle moves forward and how cells continue to live, you want to look at, first of all, what other genes play a role with this way of developing, what other genes are important for this development. And also, now that we know its role in development with J and K, how does it play a really important role in redevelopment? And that's kind of what my IRT group is looking at in the lab. And so, also something else that's interesting is that once we start gaining this ability to regrow cells, or this ability to kind of target areas of regeneration in zebrafish, and then have this ability to potentially do it in humans, how far is too far? Do we risk creating the next Frankenstein based off of continually regenerating and regenerating cells? And so, is there a limit to where we research this field? Yeah. Thank you guys so much. Thank you, Mrs. Jerko and Kelly Goins, Dr. Kirkhart, Matt Stanton, the ladies of the lateral line, and Journal Club team, my family, hi Allie, and all of you. Does anyone have any questions? Cool.
So next week we have Mr. Sullivan presenting on a new way to use gravitational waves to learn more about astrological phenomena like black holes, and to learn more about our universe. So go to that.
Not next week.
3 weeks, in 2 weeks.
Yeah.
Yeah. Remember, go to that.
So if you're in journal club, stick around after.
Yeah, Katherine.
Oh, that's